Hello. Okay, so my name's Kelsey. Going over the individual group work for this week. Um, you'll have to forgive the noise in the background. That is my family. So for question one asks about a cross that were to be performed, assuming that all the genes undergo independent assortment. The first question, or 1A, asks what portion of the offspring is expected to have the genotype of capital A, lowercase a, capital B, lowercase c, capital D, lowercase e. So for that one, there's a 1 in 128th chance of that occurring. And here's the work for it. Is I have right here, actually up here, I did the crosses, since they independently assort, that A's go together, and then these are the probability that the A will be with one of these other pairs. And so I did that for all of them. And then here is the genotypes that are asked in each question. So question A, or part A, part B, part C, and part D of question 1. I have the what's asked, and then I put all of the probabilities. So like, for example, capital A, lowercase a is a 1 in 2 chance of occurring. And so with that, I put it down here along with each of the corresponding letters, giving the 1 in 128th chance of this being a genotype that would occur in this situation. So the following one is 256. This one doesn't work because there is no little d's available. So it makes it kind of interesting. But so with, oh, where's my finger? There it is. So with this one, it just doesn't work. It would not exist. Or there could be like a mutation or something. Or a null, I don't know. Anyway. So the last one is down here, and there's a 1 in 32 chance that this would occur in this situation. So the next area is filling in blanks. So I have all the words filled out. Part A asked, paired homologous chromosomes are sometimes called blank or blank. So I just said that they were dominant or recessive because homologous pairs are the same. So whether they be big or capital letters or for both of them or lowercase for both, they would either be, I believe they'd be called either dominant or recessive for that fact. Part B, the structure that connects the centromere to the microtubules of the mitotic spindle is called a kinetophore. However one may say it, but that's what I'm going with. Part C says a diploid fruit fly has 4 pairs of chromosomes. A fruit fly cell in prophase 1 has, my answer is 8 sister chromatids, while a fruit fly cell in metaphase 2 has 2 sister chromatids. The way I just figured that if it has 4, the chromatids are the individuals, if I remember right. I've been struggling with remembering which chromosomes are chromatids, but that's what I'm guessing. An educated guess. Part D, a monohybrid cross which is the filled in blank, is a type of genetic cross that can determine if a purple flowered pea plant has the capital P's or homozygous dominant or heterozygous genotype. And hopefully that's right. I'm not completely positive. I thought about putting a Punnett square because that's how I would determine if it was one or the other, but this is like the process of it, I think. So I just continued with that, and I'll find out later. E says a non-functional allele is sometimes called, and I have to look, null alleles. While a partially functional allele is called a gain of function allele. So null is just that it's there, but it doesn't do anything, thus the non-functionalness of it. With the gain of function, I wasn't quite sure if it was this or there's another one that I can't think of what it's called all of a sudden. But this one is more or less, it's there, but doesn't have anything happen. Whereas the other possible thing I could put for the second space in part E is the one where it's more or less, it doesn't have, it just doesn't exist. Anyway, so I'm sticking with this one. Part F says Mendel's law of blank involves two traits. So I, saw that it said on the PowerPoint that it's independent assortment involving two traits, whereas Mendel's law of segregation involves a single trait, which kind of makes sense because if you're segregating something, you're separating. So a single trait would be by itself. So that's what I did for those. Question 3, which is very sloppy, and I'm not positive about this to a great degree. But so part A asks, so we have a genotype, so the capital T, capital T, capital R, lowercase r, capital Y, lowercase y, that right there. We're assuming that all of these assort independently. And so for the first question, or part A, asks to diagram all possible homologous chromosome arrangements and to indicate which is where. I started to do it and then realized that the way I was doing it just results in the same way. So I may have written this wrong, but it may be just for each, like that line and the chromatids, I think, the individual chromosomes. Oh, chromosomes, there we go, okay. There'd be 2 of them, so I think I need to figure out the big T with big T, big R with little r, big Y with little y, and then do the same down here with big Y, little y, and then figure out all the ways that works. But for the sake of time, I moved on. Part B says how many tetrads, oh, wait, sorry, wrong question. Indicate all the possible gamete genotypes that can be produced in this organism at the conclusion of meiosis. 2. And so I just took all the chromosomes and just wrote what each one would be up above. And I think that looks right. But I diagrammed it and I indicated, so I'm calling that one good. So then part C is asking about how many sister chromatids are found per cell during metaphase 1. And so I struggled, I went back and forth, but I'm going with 6, if I can remember that chromosomes. Oh, chromatids. I guess it would be 12. Anyway, I get confused because I can't keep them straight. And you can read what I wrote. So moving on. Part D asks about how many chromosomes are found per cell, but during metaphase 2. So I guess that there would be 3 just because you more or less take this and divide it in half, and that's what ends up being here. Yeah, because you've gone from haploid cells, diploid cells to haploid cells, so you have to find what's half of a diploid, if that makes sense. So that's what I did there. So then on to question 4. These are more like parts of a question. So this one's talking about horse chromosomes. And for the first one, it says how many chromosomes are present per horse cell at the conclusion of meiosis 1. And I said that there would be 16 chromosomes because you're taking the 32 and you're dividing it in half after you finish meiosis 1. So haploid cells. Part B is asking about tetrads, which I wasn't quite sure what they were, but I'm assuming that they're the pairs of chromosomes. And so at the end of prophase, or no, sorry, pardon me, during prophase 1, there'd be 32 tetrads. But correct me if I'm wrong. Part C says how many sister chromatids are present in a horse cell during prophase 1 of meiosis. Since it's asking for chromatids, I guess I should divide this in half because I was going for chromosomes in the end because I was confusing the two words. So this would end up being 32 chromosomes. Wait. Oh, chromatids. It's asking for chromatids. So there'd be 32 chromatids because those are the pairs of chromosomes. Okay. I'm going to fix this later or after. So part D, how many sister chromatids are present during or per horse cell during metaphase 2 of meiosis? So I just guessed 32. How many bivalents are present per horse cell of anaphase 2 of meiosis? I kind of thought it would be 0 during anaphase 2 because they're kind of all separated. So there wouldn't be bivalents because those are like the pairs of things. So I'm guessing 0. I don't know if that's right, but it sounds pretty good. All right, so on to D, and I'm going to try to make this work out. So, okay. So part 5, or question 5 is what I should say. It's asking, it gives an example of pea plants, gives the phenotypes of each allele, and then asks for specifics. So like the first question asks for an offspring that will be tall, round, green, or terminal, and the possibility of it happening. So that's what the work is right here. I just did the question. I wrote down and then went through and wrote all the possible ways that this could be. And so then I just took the math and I just multiplied it. And since there's 2 options, I put both probabilities, and thus giving us a 1 in 256 or 256 chance that this would occur, as I wrote down here. So then part B, right here, asks for the offspring plant with this genotype. And so once again, I took all the ratios that are up here, put those down below, and ended up with a 1 in 32 chance that it would be this way. Part C, once again, it asks for another one, but states, instead of being specific like this one is, it asks for all the heterozygous for all 4 genes. So whether that it'd be just, they'd be both different. So it'd be a big T, little t, since it's hetero, which means different. So I took once again, all of these things up here. I took each one that applied, wrote it down, calculated, and got 1/16. And then part D, which is on the next page, so forgive me one moment as I throw pages, is asking for a dwarf wrinkled yellow and axial seed. I think it's a pea plant, yeah. And so once again, took all the stuff up here and put it down to what applied to get this phenotype. And so it ended up being 1/64th of a chance to get that. So I hope that made sense. If not, I hope to learn from everyone, and I hope you have a good night. Bye.